Hey guys, we are going to continue going over monohybrid crosses, and then eventually lead into dihybrid crosses. But before we get into that, I just want to remind everybody a little bit about Gregor Mendel, who he was and what he did. So I'm bringing this back because some of the textbook questions that you guys had last week, everybody's answers weren't the greatest, and I'm not sure if it's because we're approaching the end of the school year and people are kind of just getting lazy, or we're just not reading and we're only skimming through this stuff. But if you have interest in taking AP biology, this is one of the bigger chapters, so this is going to be on there. So please try to continue reading and looking for answers. They come right out of the textbook. When we do this. So Gregor Mendel was and is still known as the father of genetics. He worked with pea plants. The reason he used pea plants is because they're easy to breed and they develop results quickly. And he was able to see what he wanted to see with the 7 traits. He came up with some laws that we still use up until this day, where he talks about the passing on of traits. One thing you should know that he was a religious monk, right? He was an Australian monk, but he also worked with science, which is something that you don't see very often. Science and religion clash more than you can think. But he was able to work with both. So the law of segregation, before we get into that, I just want to go over vocabulary words very quickly. When we talk about a dominant allele is the one that you usually see. It's the one that takes over. So it's the one that is more common, and it is usually represented by a capital letter. When we talk about a recessive allele, it's one that you don't see unless it is homozygous recessive. So they both have to be lowercase letters, and then you will see it. So during the law of segregation, during the formation of the gametes, which is your egg or sperm cells, the two alleles responsible for a trait will separate from each other, and then they will recombine at fertilization, making the genotype for the offspring. So what does that mean? It means that you will inherit one homologous chromosome from your mother and one homologous chromosome from your father. They will pair up and mix up. We call it crossover. And so that you will always result in 50% of mom and 50% of dad. Even though you may not see it, it is still there in your genotypes. So you may not physically see it in your phenotypes, but you know that it is there in your genotypes. So if you only inherit one of the dominant traits, you will automatically see that in your phenotype. So for example, brown eyes versus blue eyes, as long as you inherit an allele for brown eyes, you will automatically see brown eyes because it is the dominant trait. So law of segregation happens in the organism making the gametes. They pass on half their traits. And in this situation, we talk about parents, mom and dad. And so you will be 50-50 of each of them, even if you may not see it always. So applying the law of segregation, I just want to do a little recap of Punnett squares. Some people did really good. A few people still struggled a little bit. So over here, the 2 traits we're looking at is being tall versus short. So in this situation, being tall is dominant, being short is recessive. So tall is going to be represented by a capital T. Short is going to be represented by a lowercase t. In this situation, the mom and dad are both heterozygous, meaning they have one capital and one lowercase t. We separate those, and those will go, these letters will represent the sex cells or the genotypes, and they will go outside the Punnett square. And then inside the Punnett square, you will combine them. So for example, this T will cross over with this capital T, and you will have capital here. This capital T will cross with this lowercase t, and you will have capital lowercase, same situation here. And then these 2 little t's will cross over, and you will have 2 little t's. So if I'm looking at the phenotype only, this is a 3-to-1 ratio, meaning you have 75% chances of being tall and a 25% chance of being short. How do I know this? Because there are 3 capital T's, which tell me automatically that I will be tall. These 2 are heterozygous. These 2 are homozygous. So if you are still struggling with a monohybrid cross, I highly suggest you go back to the previous video, and I will include that over here as well. And remember how to do this before you move on to a dihybrid cross, because a dihybrid is just going to be a little bit more complicated. So there's this thing called the law of the independent assortment. And what the law of independent assortment states is that alleles for different traits are distributed to sex cells independently of one another. In other words, the allele a gamete receives for one gene is not influenced for another gene. So whatever alleles you receive from your parents does not influence something else. So I don't know why I can't stop yawning, guys. I think it's the rain. So for example, your blue eyes are not going to affect how tall you are, or your widow's peak is not going to affect, I don't know, your hair color. So your genes are not influenced by one another, they are influenced by your parents. So sometimes sets will cross over something, sometimes they don't cross over. This is where you see all of your variation. So we use dihybrid crosses to look at two traits at the same time. So I just said this, but a dihybrid cross is a breeding experiment that tracks the inheritance of two traits. So you look at two things at once. So instead of just focusing on eye color, you can now focus on eye color and height at the same time to see what your likelihood is of getting those traits. So in this first situation, we're going to go back to pea plants. We're going to look at the seed shape and seed color. So I'm looking at two things here with the phenotype. So my alleles are going to be R and Y. So RR for seed shape, YY for seed color. So a capital R will represent a round seed, which is the dominant seed. A lowercase r is going to represent a wrinkled seed, which is recessive. I literally cannot stop yawning, guys. I'm so sorry. A capital Y, and I wish I used a different letter because you can't tell the difference between a capital and lowercase, but it's okay. A capital Y in this case represents yellow, which happens to be dominant, even though we normally think of plants being green as dominant. In this situation, it's a little bit different. And a lowercase y represents green. So again, your capitals or your dominant alleles are being round and being yellow. Your recessive alleles are being wrinkled and being green. So what you're going to do, just like how we would do in a monohybrid cross, is you write out your parent allele. So in this situation, both parents are heterozygous. So they have one capital letter, one lowercase letter, one capital letter, one lowercase letter. Same thing here for mom. So they're heterozygous for both phenotypes. So they are round and yellow, crossed with a round and yellow plant. I keep saying mom, but I forget we're talking about plants. So this is the most common cross you'll see when you do tests in biology. If you go to AP Bio, if you do like state tests, this heterozygous cross is the most common that you will see. So you'll do the FOIL method. If you don't know the FOIL method for math, it's first, outer, inner, and last. So let me just forward up a second, sorry. So FOIL method usually goes for first, and then outer, then the two inner, and then the last one. So this is how we do it in math. I'm going to show it to you here in science. So I'm going to use my writing utensil. We'll go with blue. So first, right, is this capital R with this capital Y. So R and Y, capital Y, that's my first possible gamete combination. Then I'm going to go to outer, which is R with Y right here, lowercase. So R, lowercase y. I'm going to change up my colors. Then I'm going to go to inner, which is this inner R with this inner Y. And then I'm going to write that right here. And then outer, little r with little y. And I'm going to write that here. So I can get 4 possible combinations when I cross these together. And they're written right here. Capital R, capital Y, capital R, lowercase y, lowercase r with capital Y, and lowercase for both letters. These are all my possible gamete combinations, and they work in both sections. So if you cross these using the FOIL method, you will get 4 and 4 combinations that you can write down, and this is what you're going to use for your dihybrid cross. So just like a monohybrid cross, you are going to, I should have wrote this out for you. You're going to take those 4 letters and you're going to write them across the top, and you are going to write them across the side, just like you would in a monohybrid cross. So I am taking, and I'm just going to go back right here really quickly. I'm taking this capital R, capital Y, I'm bringing it here. I'm taking the next combination, the capital R lowercase y. I'm bringing it here. I'm taking the next combination, I'm bringing it to this third level, and I'm taking my last combination and I'm bringing it to this last level. So I did the same thing for both parents. I brought them over. And so now what you're going to do, and I'm just going to make this a little bit bigger. Nope, just messed it up. Okay, what you're going to do is you actually have to cross them. So I'm going to use my pen, and excuse my handwriting, but just like in a monohybrid cross, you are going to pair them up, and usually you pair them up according to the problem. So in this problem, we looked at seed shape with seed color. So seed shape was R, seed color was Y. So the R's will always come first, the Y's will always come second. So we'll do the same over here. So in this situation, I'm crossing this R with this R. So it's going to be two capital R's, R, R, and then I'm crossing this Y with this Y, and they're both capital Y's. So Y, Y. Oh my God, my handwriting's so bad. Now I'm crossing this with this R. So again, capital R, capital R. Oh my God, this is so hard. Now I'm crossing lowercase y with the capital Y. So capital Y with a little y. Now you cross the next one. So capital R, lowercase r. So capital R, lowercase r. I wish we were in school and I had a paper and pen. Capital Y, capital Y, capital Y, capital Y. This is so bad, guys. And then my last one, again, capital R, lowercase r, capital R, lowercase r, capital Y, lowercase y. So you basically have to take some time, read each one. So now I'm doing this. So I shouldn't have covered it, but so now I'm going to grab just another color. I'm going to take this row and I'm going to, or this column, and I'm going to go across with this row. So now I have, again, two capital R's, and then a capital Y, and a lowercase y, and then I'll do one more. I have a capital R, and two lowercase y's. Oh my God, that's like a V, okay? Eventually, you're going to fill out your entire cross. So I already did that, and I typed it because, you know, this is horrible. So if you, oh man, this setting messed up the spacing, but it's okay. So if you look at this, I've crossed all of my alleles here. So we did the first column together, but then we stopped here. So capital R, lowercase y, lowercase I, capital R, lowercase r, big Y, little y. So over here, when we do dihybrid crosses, we don't really count the letters up. We don't count the genotypes up. More so, we are looking at the phenotypes. Because what matters here is what they look like, right? We care what the pea plants look like. So the 4 alleles that we had, and I wrote them out here because if I sat here to write them, it would take forever, is dominant for both traits. So that would mean round and yellow. Dominant for seed shape. So that would be dominant would be round. Recessive for the color, which is green. Recessive for the shape, which is wrinkled. Dominant for the color, which is round. And then recessive growth would be wrinkled and green. So we need to figure out how many of these 4 possible combinations do our pea plants suggest. So you have to count them. So we'll start with the first one. How many plants here are both round and yellow? Well, in order to be round, you have to be a capital R. And in order to be yellow, you have to be capital Y. So this one, this one, this one, ignore this one. So I'm just looking for anything with a capital R and a capital Y. Not this one, right here, nope, yes, nope. Okay, so how many is that? Let's count it up. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's 9 right here. So again, all I did was I looked for anything with a capital R and a capital Y. Now I'm looking for a dominant round, so I'm looking for a capital R, but I'm looking for 2 little y's. So I got, I'm gonna use a different color. I'm gonna use, I don't know, like a hot pink, and I'll do a circle. So right here, anything with a capital R, lowercase right there, right here. No, no, no. So over here, I got 3. So again, this is out of 16. So 9 out of 16, 3 out of 16. Now my next one, I am looking for a lowercase r in both, and I'm looking for at least one capital Y. So I'm only left with 4 squares. So this one, obviously, this one, and this one all have a capital Y and lowercase. So again, 3 out of 16. And now I'm looking for both lowercase letters, and there's only one right here. So that's 1. So in the end, your ratio is going to be 9 to 3 to 3 to 1. So this is the most common type of example you will see. If both parents are heterozygous alleles, you will always have a 9 to 3 to 3 1 ratio of your thing. So here, this is way more cleaner. So when you do this, so round and yellow, there are 9 plants that are going to come out that are not round and yellow. There are 3 plants that are going to come out round, but will be green in color. 3 that are going to come out wrinkled, but yellow in color. And then there's only going to be 1 possibility, you're recessive, of being both wrinkled and green. So you'll see the dominant trait is the one that is more visible. Recessive is the one that is less visible. So in your textbook and online, when you look this up, right, round and yellow right here, all of these are round and yellow. So 9. Round and green, you have 3 right here, 1, 2, and 3. Wrinkled and yellow, 1, 2, and 3 right here. And then wrinkled and green is your last option right here. So this is an example of a dihybrid cross. You guys are going to be doing practice problems for this today as well. I'd like you to do it on a piece of paper and then email it to me or attach it so that I can see where you are struggling. Thank you.